users. So today I am going to discuss about how membrane mechanical properties are affecting the transport process. So to start with, transport against the along the cell membrane is very important for homeostasis for the functioning of cells, and a cell have different mechanism to transport material across it. For example, if the molecule is very small, it can just pass through diffusion. Now, if the molecule is large enough that it cannot diffuse, then cells have different mechanism. They open up some channels through which molecule can pass based on their concentration gradient. But if cells need to pass molecules against the concentration gradient, then also cells have different mechanisms. They have pumps, they have carrier proteins. Now this carrier proteins is very interesting. So they go through conformational changes. They form closed conformation, open conformation, and they do it cyclically. And in one cycle, some material pass the cell membrane, and this is a directed motion. It doesn't care about where the gradient is higher or lower. It directly, in one direction, it passes the molecule. Now, one such kind of this carrier protein is called ABC transporter. This stands for ATP binding cassette. So they have some domain at which ATP can bind. They go through conformation changes. ATP will hydrolyze, and at the end, some molecule will be transferred. Now, one such ABC transporter is BMRA, which is very important, and because these are also the multi-drug resistant proteins. So bacteria actually develop multi-drug resistance, and they use these kind of proteins to throw out drugs that are inside their cell body. And experiment by our collaborator is performed on this protein, and they use some liposomes. These are artificial cell membranes, so there are no other component than the lipid bilayer only. There are no protein, no cytoskeleton. And so to understand the mechanism a bit, just concentrate on this schematics. So this is the open conformation where a substrate can bind to the protein, to this transporter. Then thermal fluctuation can make it to a closed conformation. ATP binding can stabilize this closed conformation, and at the end this substrate will transfer and ATP will hydrolyze and then will go back to this conformation. And in this one cycle, one ATP will be hydrolyzed and one or more molecule will be crossing the cell membrane. Now the experiment on two population, one large liposome and one small liposome. Our collaborators noticed that there are very different results what they obtained. So the closed fraction of closed population is different for large vesicle and small vesicle, and the ATP activity, which is defined at ATP hydrolysis rate per unit time per unit volume, this is also different. So somehow the mechanical properties of this liposome, which is the curvature here, is affecting the result. And our aim is to understand this using very simple model. So the model that we consider is very simple. So we have arc that we rotate around the z-axis, and it will form a closed membrane. So this is axisymmetric model. So the parameter S is 0 here and L here, and for any value of S I can define these three parameters: slope on that point, radius, which is the distance from this axis, and then horizontal is in Z. And this open space, the dashed line, is the space where we can insert protein of size A. Now what are the energies? So the first energy may be the area, a surface energy, but the population is such that there are no addition or subtraction of lipid molecules. So once they are constructed, they are like constant area. So area is, or surface strength is not playing a role, but these proteins are hydrophilic. So whenever conformation changes, they deform, and they also deform the membrane because these are hydrophilic. Membranes have hydrophobic part. So if they deform, then they try to maintain this contact angle 90 degrees so that to minimize the hydrophobic to hydrophilic contact. So bending energy is playing a role. Now what will be the form of it? Is it just the mean curvature square? It will not be so, because for such a small liposomes, there are asymmetry between the number of lipids of inside and outside layers. Thereby there will be some spontaneous curvature, and we can assume that this spontaneous curvature is like that when they are perfectly sphere, the energy is minimum. That's all. So we can calculate the radius. So if area A0 is the area and I insert a protein of size A, then the new area keeping it spherical will be, so the new curvature will be this, keeping it spherical. So this value we can use as a spontaneous curvature. So we have the energy, and then the aim is to calculate the membrane shape, minimizing this energy. So physical system has a tendency to minimize the energy. So given the boundary condition, the shape at which the energy will be minimized will be the one that is relevant physically. So this process is very like well established. We have to find out the Lagrangian, use the constant, area constant, geometrical constant, and then we have to write down Euler-Lagrange equation for each variable. These are the equations, and the boundary conditions are also important. They are imposed by the protein size, slope of the proteins, and then we have to solve it numerically. So one such solution will look something like this. So minimum energy when the shape is perfectly sphere, and if I am deforming whether positively or negatively, in each case the energy will go up, and this is slope, this x-axis is slope, positive or negative, and within a small deformation I can fit a quadratic function here to the bending energy. So the bending energy scales as theta minus theta 0 square. So this is saying you how much the shape is deviating from a sphere, and this prefactor alpha scales as curvature square, where is the protein size sitting here. So I can write down the energy in this way, change in energy in this way. So now our interest is to find out the change in energy from two conformations. One is the open, another is the closed. That's why I find out the change in energy from open to close, and this may or may not be positive. But given the value of theta open and theta close that our experimental collaborators provided us, del E turns out to be positive. So open angle is energetically favorable. In order to go from open to close, the system has to cross a barrier of delta E, and the barrier becomes larger for smaller liposomes. So this is the model conclusion, and with this conclusion we can explain all the data that I showed you in the first slide. So we can do a little bit of statistical analysis. So open population and closed population. K1 is the thermal fluctuation that will take open to close, and K2 is this rate that involves ATP hydrolysis. And in the steady state, I can write down the ratio of this population as the ratio of these rates. So in experiment, there are two kinds of ATP. One is the hydrolyzable, and another is the AMP PMP, which is non-hydrolyzable ATP. So they bind with the ATP. Yes, 1 minute. Bind and then they unbind without hydrolysis. So this is like an equilibrium process. This is like a non-equilibrium. These rates are different, but I can write down for both the cases this rate, and then these are the experimental data, and I can also calculate the fraction of open population that experimentally is measured for both the cases, and also the ATP activity, which is the ATP hydrolysis rate per unit time per unit volume. And then I can fit, unfortunately or fortunately we have 2 points only, it's very easy to fit. So these are the experimental values provided by our experimental collaborators. Fitting parameter turns out to be 16, which the order is quite good. So in summary, we understand that BMRA proteins are mechanosensitive. By the way, this is the first experimental study to reveal that this protein is mechanosensitive. And using simple model, we also understand qualitatively the mechanosensitivity. And I would like to thank our collaborators and funding agencies, and thank you all for your kind attention.
Questions?
So, hi. I just wanted to know how this is moving forward, that your ATP molecules.
What are moving
with
forward?
that, your ATP molecules are there, like particles are there. They are moving forward, right? They are bending and stretching the angle and then moving forward, right?
Okay, so we are not doing any kind of dynamics. We are interested in only shape of the vesicle. Okay, when for two different conformation, open or closed, what is the shape of the vesicle? That's all.
Okay.
What ATP is doing, what are the dynamics of ATP, we are not interested in the model, at least.
Okay. Thank you.
But a two-state process like open
This is
to
a two-step process.
Yes, but a two-state process is always in equilibrium.
Yes, yes,
So what is
you are right.
non-equilibrium there?
So there is no difference actually.
No, but you also talked about one
Yes,
non-equilibrium
in
and
reality, one involve ATP hydrolysis, one doesn't involve ATP hydrolysis.
what is non-equilibrium in two-state process?
So if I have, so in two, like in terms of model, there are no. But in reality, there are ATP hydrolysis involved. That's why in reality, one is non-equilibrium. Always detailed balance will be satisfied in this case, of course.
So why do you have only 2 points?
2 points. These are the two population, experimental population, large vesicle, small vesicle. Unfortunately, they don't have any third point. So our aim is to fit it. Yeah,
That's actually my confusion. With the 2 points now.
I can fit elephant. But that's from a experimental side. We don't have anything to do. KBT is, yes. The order is correct. The order is correct. So numerically, I will not say, but order is correct. Order is correct. But this is very simple model. If the order is correct, then it is a successful model.
Okay, let's thanks to the speaker.